Hello everyone and welcome back to BioByte Lab. In today's video, we are starting one of the most important topics in research and biostatistics, and that is sample size calculation. Whether you are conducting a survey, a clinical trial, laboratory research, or any biomedical study, one of the first questions you will face is how many participants or samples do I actually need? Second question, why sample size is important? So imagine you collect data from only 10 people. Your study might miss real differences simply because the sample is too small. Now imagine collecting data from 5,000 people when only 300 were actually needed. That means you have spent extra time, money, and resources unnecessarily. So, choosing the correct sample size helps us balance first scientific accuracy, second statistical reliability, third time, fourth cost, fifth critical consideration. So, what is sample size? Sample size simply means the number of observations, participants, patients, samples, or experimental units included in a study. For example, one hundred 50 patients in a clinical trial. 300 survey responses, 80 blood samples, and 50 lab experiments. These numbers represent the study sample size. So what happens if the sample size is too small? A small sample size can lead to 1. Low statistical power, second, unreliable conclusions, third, missing true effects, fourth, large confidence intervals, fifth, increased risk of false negative results. In simpler words, Even if a real difference exists, your study may fail to detect it. So the next question is, what happens if the sample size is too large? A very large sample size can also be problematic. It may waste your resources, it may increase study costs, or it may take more time, or it may expose unnecessary unnecessary uh, participants in clinical research. Fifth, it may detect statistical, statistically significant but clinically meaning, meaningless differences. So it means bigger is not always better. So sample size is not just one formula. Many beginners think there is a single sample size formula. Actually, the calculation depends on your study design. Different studies require different methods. For example, let me write it down. So, different studies require different methods of sample size calculation. For example, estimating estimating a population proportion, comparing two proportions, comparing two means, paired studies, correlation studies, survival analysis, regression models, diagnostic accuracy studies. So each has its own statistical assumptions and formulas. So the next question is, what determines sample size? Let me write it down. What determines sample size? So there are several factors that influence the required sample size. These factors are: number one, confidence, uh, confidence level; second, margin of error; third, expected prevalence; or proportion. Fourth, standard deviation. Fifth, effect size. Sixth, statistical power. Seventh, significance level, which is alpha, also known as alpha basically. So we will explain each of these in separate videos before performing calculations. The next question is, why calculate simple size in R? Many researchers use online calculators. They are quick but often limited. So R provides much greater flexibility because it allows you to calculate sample size for different study designs, customize parameters, reproduce your analysis, generate publication quality outputs, and perform advanced power analysis. Most importantly, R is free, open source, and widely used in research worldwide. So what will you learn in this series? Over the next videos, we will cover basic statistical concepts needed for sample size, installing required R packages, sample size for prevalence studies, sample size for comparing means, sample size for comparing proportions, power analysis, effect size, practical examples from biomedical research. By the end of this mini series, you will be able to calculate sample size confidently for your own research project. In the next video, we will begin with statistical concepts that every researcher must understand before calculating sample size in R. If you found this introduction helpful, don't forget to like the video, subscribe to BioByte Lab, and turn on notifications so you don't miss the upcoming tutorials. Let's dive into the world of research statistics with R. Till then, happy learning.